So thank you very much for the kind introduction. It's really a pleasure for me to be here. And before I start my presentation, I just want to thank the Hilsi and Delia that is sitting in front of here for the nice and kind invitation. I mean, I always wanted to attend this meeting. This is my first time. So when I received the invitation, actually, I was very pleased because, you know, it was like a kind of desire that becomes true. But then I saw the title of the session, I mean, zero tolerance, and coming from Europe, where we actually have limits, you know, it was a little bit, I don't know, let's say, impacting. And then, of course, when I went to the proposed title, I felt a little bit more confident because whole genome sequencing is in the area where we are actually working, not directly, but trying at least to implement some of the information that we get from the whole genome sequencing to make our foods more safe. So today I'm going to show you some views, some work that has been done that is actually very promising towards the use of these approaches in order to improve safety of foods. And then I'm going to give you some perspectives. I mean, probably at the end of the session, you will go out with more questions than answers. But it's my role to do that because I think that we need to be in a position to understand what are the possibilities that new methodologies that can give us. And so we don't have to give up because probably the scenario is too complex at this moment, okay? So, of course, I believe that everybody in this room, when he attended a session on whole genome sequencing, saw a graph like that. I couldn't resist not to put it, really, because this is really putting into the context the sequencing capabilities and the exploitation of these methodologies. So as you can see, practically the cost of sequencing has reduced has a huge jump, a huge decrease. And if you take into consideration, for instance, right now, I mean, I was a couple of days ago in my room trying to shape the presentation. I would not say to prepare it, but to shape the presentation. I just did the search, and as you can see, there is a huge amount of information for specific organisms that are, you know, the very known ones. But you can see that there is also some kind of increase in terms of microorganisms that are becoming really relevant by the point of view of food safety. For instance, you have already 47 genomes for Archobacter. Of course, Listeria, Salmonella, Campylobacter are very well-known foodborne pathogens, so there is an interest from scientists to look into it. And then if you just plot the number of the sequences in the last 20 years or 15 years, you can see that the huge increase has actually took place when the cost of the sequencing was reduced. So as if, you know, food scientists, in any case, they started to approach this methodology when the cost was very low. And of course, this is absolutely understandable and reasonable. But the other thing is that we have also a very high speed of sequencing. So if you take into consideration the speed of sequencing, considering the kilobase per day, you can see that practically in the last 5 years, we have experienced what is called the third generation sequencing area, in which you can start to go for very large pieces of DNA to be sequenced. I mean, there are different technologies. Probably the one that is the most exciting at the moment is this mini-ion Oxford nanopore that exploiting nanopore technology, you can actually have portable devices that after a 15 minutes preparation of the sample, you can get your sequences right away on the spot without having to transfer samples from fields to lab with all the problems that we know. So we are really in an exciting moment, in an exciting scenario. However, I see a high risk, and this is something that we start to take into consideration, that we are going to get overloaded with sequencing data. So we became very good in technology by the point of view for the sequencing. We start to become good in terms of handling the data. the next challenge, I think, is going to be how we are going to translate the outcomes from the sequencing to the real world. Because I'm a food microbiologist, so I want to see things applied. So if we have 1 million sequences or 10 million sequences of Listeria monocytogenes available, but we don't have a clear message to send to the food producers, then I think that we have a problem. So my role today is to take into consideration how next generation sequencing, or sorry, whole genome sequencing is actually going to improve or to impact on the risk assessments and regulations. As I'm going to show you a little bit later, we have had in Europe a workshop where we try to explore, we try to discuss or to start a discussion on how these methodologies could be integrated into microbiology risk assessment, microbiological risk assessment. This is a first paper that has been produced by the working group dealing with whole genome sequencing, and the main outcomes of this group were that the whole genome sequencing can really give you a nice insight on foodborne pathogen surveillance, can be used for understanding reservoirs and delineating transmission routes. This is something already said. But of course, you have also a very good possibility in contributions to the risk assessment and upgrading the hazard identification. So I'm going to go through these 3 points a little bit, just give you some insight. Of course, there are not inclusive insights because the examples that you can find in the literature are huge in a sense that there is a large number of papers that have been already published on this specific subject. Here we talked already about how the whole genome sequencing can give you like a real burst in terms of understanding the epidemiology. So you have just 2 papers of 2017 published in July and also in July and in January about multi-country outbreak of Salmonella enteritidis. This is something that has happened in Europe, looking at the distribution of Salmonella strains that were responsible for outbreaks both in England but also in Spain. So you have also like a transnational migration, let's say, of these microorganisms. And then the other one is again related to Salmonella and in Australia. So this, I think, that is something clear to everybody. Being able to look into the genome of the different microorganisms, we can really understand how they are related to each other, if there is the same strain that is responsible for the outbreak in different countries. So we have a very good tool that is for sure much more, let's say, deep in terms of the knowledge that you can get compared to what we were used to exploit in the past. Related to understanding of reservoirs, again, there are a number of papers that they start already to take into consideration this aspect. Here there are 2 examples. We talked a lot already these 2 days about the ice cream case here in the United States. But then you have also cases of tracking pathogenic microorganisms in plants. So in the study on the bottom, practically what they did was to use whole genome sequencing to understand how the pathogenic strains of Listeria monocytogenes in particular were detected in the different parts of the plant, and they were also able to understand a little bit how they were moving and what was the cause for them to move. But one of the aspects that I want to underline, and let me spend a couple of minutes on that, is a kind of translation of what has been done in this study that was produced by the group of David Mills in California, where by using modern technologies, and here we are not talking about whole genome sequencing, but the outcomes can really be translated to whole genome sequencing when we talk about foodborne pathogens, is actually taking a very detailed picture of what is going on in your plant. So here, what they did was to apply 16S high-throughput sequencing for a brewery plant. This is a nice representation of the plant with the different sectors, fermentation, bottling, and everything else. And what they have done was to take samples from each part of the plant and create these kind of heat maps where you can really understand how contamination occurs. So here you can see practically the source. So these are grains, hops and yeasts, and the darker the color, the higher the presence of the specific microorganisms. So you can see how the contamination is changing in the plant. You can see also how it's changing in during the year in the different seasons. And here is the last example of the human skin, for instance. So how human is actually contributing to the microbiota of the plant. And so think about how this can be implemented for the foodborne pathogens, let's say, distribution. So you can take samples, you can get your pathogen, if it is present, out of the sample. You can do whole genome sequencing. Actually, you can track those microorganisms in your plant. And this, of course, it is very, very important because you will get an information on how the pathogen gets into your plant, and then, of course, you can do control measures in order to avoid this to happen. The last point is the contribution to risk assessment. So here we are talking about risk assessments. I'm not a risk assessor, so I'm a food microbiologist. Let's say I'm like an ecologist or microbial ecologist. So I'm a little bit, out of context, or in any case, I'm not an expert on that. But I think that in any case, we need to think multidisciplinary. So the future is going to be the integration of the different disciplines in order to face or to the challenge, of what we are talking about. So this is a nice example on how whole genome sequencing can actually give you an insight in terms of hazard identification. So in this paper, a group from Netherlands, what they have done was to do whole genome sequencing of a number of enterohemorrhagic E. coli. So by looking at the sequencing data, pure sequencing data, they really confirmed the genetic lineages. So going into the DNA sequence, they could identify populations, and they could identify, or they could connect, let's say, the populations with the genetic lineages. This was not enough, of course, because if we look into the difference at the genetic level, you stop there. You don't know how the microorganism is actually behaving, or how the microorganism can actually be potentially pathogenic. So what they did was to do in vitro studies to understand the adhesion and the invasion, and they correlate the genomic data with the physiological data. And so they came out with these rather impressive graphs where every single line represents SNPs present in the genomes of the microorganisms. In the first graph on your left, you have practically the result of the SNPs without taking into consideration without the correction, of the population structure. And in this case, practically you have an overestimation of potential significant SNPs that can make a difference between, or that could be related with the difference in the physiological manifestation, let's say, of the pathogenicity. And in the other case, they took into consideration also the correction of the population structure. So taking into consideration genomic data and physiological data. And as you can see, only one SNP could be considered as relevant to explain the differences at the physiological data. So this was proposed to be a good potential biomarker. So in a population of different strains of enteromorrhagic E. coli, you could really just focus, or let's say, focus at least on this biomarker to understand also the potential that this strain can have at pathogenic level. But now I want to jump a little bit, and I want to take you somehow in the future. So let's get on board of the machine of the future, let's say, of the movie, you know, and just try to address it or to approach this subject in a different way. We have genetic data. As I told you, we don't know, if we stop at genetic data, we don't know anything about the behavior of the microorganism. So if we want to understand, or if we want to have an insight on how the microorganism is actually behaving in a specific environment, necessarily you have to go to other indices of activity. One can be proteins, of course. The other one can be RNA. So I believe that one of the pioneering studies taking into consideration and demonstrating how microorganisms, and here specifically you were talking about Listeria monocytogenes, is actually reacting to a specific environment, was the paper published by Kosar at the beginning of the 2000s, related to Listeria monocytogenes. So if Listeria monocytogenes is present in different environments, it actually expresses different genes, and is moving its transcriptome based on the inputs coming from the environment. So here you have different cases. You have growth at 30 degrees, stationary phase, hypoxia, cultivation in blood, cultivation in the intestinal environment. And so as you can see, practically the number of the genes are really, really different based on the environment. And if we categorize a little bit the number of genes, here it's going to be practically impossible for you to see, but just let me highlight how the number of the genes that are related with pathogenicity, and so virulence markers, are actually going up or going down based on the environment that Listeria monocytogenes is finding. So as the title says, practically Listeria can be considered or can act as a saprophyte or as a pathogen based also on the environment. And so think about how those results can be actually translated when we are talking about food microbiology and our foods that contains this kind of microorganism. This is something that we have tried somehow to address. This is a paper that we have prepared some years ago in a big European project that was addressing the Contra strategies for pathogenic microorganisms in food. And practically what we have proposed is that as a flow, we have to understand a little bit better what is going on inside of our cells in terms of genomic, but also transcriptomic and proteomic data. We need to integrate those data with bioinformatics and also chemometrics. It is extremely important to have phenotypic data because this is really a critical point. I mean, yesterday I attended some sessions where they were saying we have this huge amount of data already available by the genome sequencing point of view. So how are we going to exploit them? Well, if we don't have any kind of phenotypic data, it is going to be very, very, very hard to exploit them. We can exploit them by the point of view, as I was telling you before, because we can do source tracking, we can do surveillance much better. However, how we can integrate or how we can use them for risk assessment, then I see it a little bit more difficult. And so, as you can see then, there is practically the last part that is related to interpretation of the data, new hypotheses that are going to be made, and of course we have to validate the experiment. So, taking into consideration these aspects, one more point that I want to contribute with is how important is the food matrix. Most of the data that at least have been generated in the last, I would say, 15, 20 years, have been related to in vitro laboratory conditions. So very few experiments have been done in real food matrices. And if you remember what I was telling you about the Listeria paper, there is a huge influence of the environment on the physiology and on the gene expression of the pathogenic microorganisms. So what we have done here in this study was just a very simple experiment, like a proof of principle, in which we took 3 different strains of Listeria monocytogenes. We simulated a food matrix. This was just, you can see, meat juices. So it was just an homogenate of meat and an homogenate of fermented sausage. And we inoculated these strains in different conditions, 4 degrees, and for 30 minutes and 48 hours. We looked at the expression with a sub-array. It was a microarray that was specifically designed, again, on the European project that I told you before. But what I want to give you as a message, as a take-home message, is that when we compared the actual gene expression of this set of genes that were all related to virulence, to adhesion, to stress response, we could actually see an overexpression of those genes in food systems compared to the in vitro laboratory conditions. So the expression of the genes were normalized towards the growth on BHI, 37 degrees, and you can see that most of the genes, at least there is a huge amount on top and also on the bottom, are actually highly expressed, upregulated. So there is a problem somehow, or there is, let me not just call it problem, but we need to take into consideration that doing things in the laboratory conditions and doing things in the real world are actually completely different and can give you completely different things. We have tried somehow to integrate or to do some kind of integration related to expression data and risk assessment. So what we have done was to actually look at the expression of Listeria monocytogenes in 2 different sausages. One, the cacciatore, is the fast acidifying. 3, wow, is a little bit different from my count anyway. So is a fast acidifying. The felino is a slow acidifying. So we looked for I'm going to go fast. We looked for the expression of different genes by quantitative PCR, and then we could see, a kind of differences in the expression. Several genes were differentially expressed, but then when we integrated the data of gene expression with the physiological data and actually with the quantification of the persistence of these strains in fermented sausages, we could actually create these nice maps in which we could identify genes that were very closely related to inactivation. So in this case, as I said before, we could really identify biomarkers that could be used as signals for inactivation of the microorganism. This is what I discussed about. So last year, what we have done in collaboration with the ILSI in Europe, IAFP and the ICFMH, we organized a workshop related to the next generation mRNA. We had 4 groups. Yesterday, as some of you attended, we had already a symposium on these things. I just want to give you like a fast overview of what we have done, because when we take into consideration sequencing, sequencing is a lot of stuff. And so when we take whole genome sequencing is just a very tiny part of the next generation sequencing scenario. So we need to be aware that there are a lot of potentials of these methodologies, and of course we need to start to think how these potentials can be exploited. Those are the possibilities, in my opinion. Of course, this is the result of a paper that we have produced from the working group of the metagenomics, And what it is, in my opinion, most important is actually the part down here. So from metagenomic analysis, you can actually reconstruct whole genome sequencing. So at some point, we will not need anymore to isolate our foodborne pathogens from the samples, because if the sequencing is deep enough, we will be able to get whole genomes from the metagenomes. And so this is a kind of direct whole genome sequencing without cultivation, and we know we never touched this issue yet in this symposium, but you know how hard it is to actually cultivate foodborne pathogens. And you know how many biases are related to the cultivation. And of course, from whole genome sequencing, we can go to prediction, we can go to genome scale metabolic models, and this is going to be impacting, in my personal opinion, the way we will do microbiological risk assessments. So this is just a very, a snapshot of what we have done. Again, this is going to be probably creating much more questions or doubts than giving you answers. But so we have looked into this is a metagenomic shotgun library. This is ecology, nothing new. Those here are fermented sausages without the addition of starter cultures. These are sausages with the starter culture. So it is obvious that when we add the starter culture, we see only lactobacillus and we see only staphylococcus, while in the other case, we see a lot of other microorganisms. We can create these nice networks taking into consideration the activities. So here you have the metabolites of carbohydrate, amino acids, and lipids in the spontaneous and inoculated. But what is more interesting, in my opinion, how this can be related to food safety and quantitative microbiological risk assessment. So what we have done was to look for sequences of pathogenic microorganisms. We found Listeria monocytogenes sequences. Very important to underline, those are sequences that the total sequences, so total microbiota sequences, you can see that we can go up to 70,000 sequences related to the ecology of the system. And here you have the Listeria monocytogenes sequences. So you have like a tiny, tiny, tiny part of the sequences that are actually, you know, in this overall sea of sequences related to the other microorganisms. So looking a little bit more better into Listeria monocytogenes, you can see that practically there are a lot of genes related to metabolism, to the genetic information and processes. And what is more important is that then if we go further in the second level characterization and even more into virulence factors, we find some sequences, but you can see that there are just a very few. So we have 5 sequences, for instance, of the invasion-associated secreted endopeptidase, for instance. We have some sequences, nothing for the phosphodiethyl, the phospholipase, for instance. So how do we address and how we interpret these results? I don't know. I don't know. So again, we are technologically ready. We have the pipelines and the bioinformatic tools in place. However, probably we are not ready by the point of view of the understanding and the exploiting of the results that we get. So there is some work that we need to do. So just a last input that I want to give you. We are working in a complex ecosystems. So we need to take into consideration these aspects. And in my opinion, it is very important that we need to take into consideration the whole picture. It's not like in the performances, you know, of our kids at Christmas that every single partner is looking at his kid and is taking pictures or movies of the kid. We need to have a picture of the whole system, because only in this way we will be able to understand. So just to get to the last slide, the question was how whole genome sequencing, or better next generation sequencing, is actually impacting assessment of risk and setting of standards. For the first questions, I'm rather positive. I mean, we have the potentials. Of course, we need investments, because the problem that we have right now is that we don't have much money to run these experiments. Of course, we have a lot of different obstacles that need to be overcome in the future. How they will set standards, I think that this is going to give us a lot of headaches yet, still. So I'm not positive, and I'm really convinced that this should not, at this moment, influence the setting of the standards. Listeria monocytogenes is a pathogenic microorganism. We cannot allow ourselves still to consider, or let me put it better, we cannot consider different strains within the same species. So we still need to consider Listeria monocytogenes as a single pathogenic species until when we will have a better background in order to fully address this problem. And with that, I just want to acknowledge the people that has worked with us from the DISAFA, from the graph here. It is Marius Mataragas. He's a little bit hidden, but he's the one that did a lot of these risk assessment things. And of course, all the people that have funded our research so far. Thank you very much.